Hello there, micro students. This is just a quick review on nucleotide structure and how they are used to make a really important molecule called DNA, or deoxyribonucleic acid. And so this is something you should have learned in introductory biology. You get reviewed in other classes as well. So just wanted to do a quick review for this. So nucleic acids are one of the four biological macromolecules that are used to build all living things, all living cells and organisms. And so the four macromolecules we're interested in are nucleic acids, as I previously mentioned, but included with those would be carbohydrates or sugars, fats, or also called lipids, and then proteins. And so we want to focus on the first category of these biological macromolecules that are called nucleic acids. And so nucleic acids are made up of building blocks that are called nucleotides. And so all nucleotides are going to have three parts that are going to be illustrated on this slide. And so one of them is it always contains a sugar that's in the center of the molecule. And then off to one side, there's something called a nitrogenous base, or a base for short. And then there is, off to the other side of the sugar, something called a phosphate group. And phosphate groups, there can be one phosphate group, but it can have up to three phosphate groups depending upon what type of nucleotide. And so those are the three parts of any nucleotide. And that's reflected here. So you can see here in the center of the molecule, that's the sugar. And then off to the side here, it's a nitrogenous base, or base for short. And then this one has just a single phosphate group to it. So those are the three parts. Nucleotides, we said, are used to build nucleic acid, and there are two types of nucleic acids that you have learned about. So just to review, DNA is one type of nucleic acid, but then there's also RNA. And so nucleotides are used to build both of these molecules because these are both nucleic acids. But what's going to vary is the structure of the individual nucleotides that are used to build them. So the sugar differs between the nucleotides for DNA and RNA. And so the sugar that's in the nucleotide to build DNA is called deoxyribose. And the sugar in RNA is not deoxyribose, but rather it is ribose. So that's one difference between them. The other is in the bases that exists in each of these. And so there are four different bases that can be linked to the appropriate sugar. So in the nucleotides for DNA, it's deoxyribose. That's the sugar. And so then the question is, well, what nitrogenous bases can be attached to it? And so in DNA, there are four different bases. So a G, a C, a T, or an A. And those letters are short for these different bases. So adenine is A, guanine is G, cytosine is C, and thymine is the T. The bases in the nucleotides to build RNA, three of them are the same. So there can be guanine, cytosine, and adenine, but there is no thymine base for RNA. Instead, there's a different one called uracil. So if you notice, RNA only or DNA only. So those are the really important differences of the nucleotides that can build DNA or RNA. So the structure of a nucleotide is a sugar that's often drawn as a pentagon. And then off to one side, there is a base. And then off to the other side, there's a phosphate group. So that's what one single nucleotide looks like. But to build a nucleic acid, what happens is, if you have two of these building blocks, and so they have this similar structure, what's going to happen is a bond is going to form between them. And so the bond that we're interested in here is going to form, I'm just going to draw it as a really dark line. So instead of having two separate nucleotides, you now have two linked together. And so we have what is a nucleic acid, and this bond has a name, of course, And so this is from chemistry. It's called a phosphodiester bond, is what it is. But this is what links all nucleotides together to convert them into a structure that we call nucleic acid. So this is just, these are two nucleotides linked together, but I could draw a bunch of these, which I'm not going to do, but I'll just do a third one here. So here's a third nucleotide, and then here's another phosphodiester bond. And so now what we have is what we call a growing chain or strand of a nucleic acid. Okay, so we could be looking at DNA here, we could be looking at RNA here. This is a similar structure to both. What's going to differ, of course, is what is this sugar? Is it deoxyribose or ribose? And then what are these bases? Are they A's, G's, C's, and T's, which would be for DNA, or are they A's, G's, C's, and U's for RNA? Okay, so this is a more generalized structure. One other thing that I want to bring to your attention is what we call this directionality. And so each end of a nucleic acid chain can be designated with these labels. So this says 5 prime, and this says 3 prime. And this just has to do with, it's how we name molecules within chemistry. So without going into some organic chemistry naming, just realize that this right there, this little corner, is the 3 prime carbon, and there is not another nucleotide attached to it. So this is the free 3 prime end. And then up here, this phosphate is attached to the 5 prime carbon, and there is not another nucleotide here. And so these names are just telling us which end is which. So this is the free 5 prime end, this is the free 3 prime end. Most importantly is just knowing this. If we were to draw a line to represent any nucleotide and said, if this is the 3 prime end, then we know this other end has to be the 5 prime end. And that orientation or that directionality is important for many of the processes that we're going to see in the future. Okay, so most DNA is not going to exist just as a single strand, but instead it's going to exist in what we call a double helix. And so rather than a single strand, there's actually 2 strands that interact. And knowing the structure of how these 2 strands interact is really critical for understanding processes we're going to see in the future. And so the 2 strands, I'm going to draw another one over here, but what you're going to notice is I'm actually drawing it running in the exact opposite direction. So it looks like it's upside down. So if you look at my pentagons, it's a little bit harder for me to draw. But those pentagons that's supposed to be a phosphate. That's a base, and we'll do one more just to make it similar. So there's a phosphate and there's a base. Okay. So because these are oriented, and I guess I can do this too. There's the phosphodiester bond. There's the phosphodiester bond. So now we have 2 chains of nucleic acid. And one thing to notice is that they're running in opposite directions. And so from the top, this is 5 prime to 3 prime, but this one from the top, it's 3 prime to 5 prime. And so the term we use to describe this is called anti-parallel. And that's just the direction that these 2 strands are interacting. So yes, it's a double helix, but they're not going in the same direction, they're going in opposite directions. And the other thing that's really important is there's always a certain way in which the bases are going to interact. And so we call this base pairing, or the base pair relationship. So right now I just have B's as generic, but remember what your options are for nitrogenous bases for DNA. There could be an A here, and so I'm just going to scratch this off and say, okay, here, say there was an A on this strand. Say there's a G here, and then say there's a T here. The base pairs are always as follows. If there's an A on one strand, that's always going to base pair with a T on the other. if there's a G on one strand, it's always going to base pair with a C on the other strand. So if there's an A here, the base on this corresponding second strand has to be a T, A and a T. This, I think I said was a G. I'll just make that better. If there's a G here, then you have to have a C with it. So there's the base pair, a C and a G. And because we have a T here, this would have to be an A. And so this is the structure of DNA where we can talk about the base pairs. We can talk about it being double-stranded, and not just double-stranded, but the two strands are anti-parallel. And while we talked about the phosphodiester bonds that are important in one strand, what I didn't tell you about is why these two bases pair in the way that they do. So why is it that A and T base pair and G and C base pair? It has to do with bonds that form between it. They are not as strong as those phosphodiester bonds, but these are, they're called hydrogen bonds. And so it's the hydrogen bonds that actually form and hold the bases on the complementary strands together. So I know this picture isn't great, but just to talk you through it, I figured I'd draw it. And so this last image just reinforces everything that we said previously. So notice a couple things. There, for DNA structure, there are two strands, and they are organized in what we call a double helix. So they are not, they kind of are oriented like this spiral staircase. So notice that double helix. Notice those two strands are not oriented in the exact same direction, but instead we say, oh, those are anti-parallel. And so this picture shows that as well. If we were to take this one strand right here, this purple strand, and zoom in on it, we would see what actually is making this up is this would be one nucleotide right here. So here's the sugar, here's the base, and here's the phosphate. And so that's one nucleotide. And if you look here, here's a sugar, here's a base, and here's a phosphate. And so that's a second nucleotide, but they have been linked together with that bond that I mentioned before, which is called a phosphodiester bond is what that's called. And so that's what's holding all of the nucleotides on this one chain together. But because DNA isn't just a single strand or a single chain, but instead there are two, this strand over here also held together. So this here would be also a phosphodiester bond. But what's holding this chain and this chain together are these base pairs. And so again, notice the base pairs here. You have a G pairing with a C, and then you have an A pairing with a T. So the base pair relationship is something also that's really important in the structure of DNA. Okay. And then these bonds that are holding the base pairs together are not phosphodiester, but they are hydrogen bonds. So I think that's pretty good for the structure of DNA and the building blocks that make them up called nucleotides.